Welcome to Developmental Biology. Today, we are going to explore one of the most fascinating questions in science. How does a single, seemingly uniform egg cell know exactly how to build a complex three-dimensional organism with a head, a tail, a back, and a belly? The answer lies in a remarkable mechanism called cytoplasmic determinants. By the end of this video, you will understand not just the biology behind this process, but also the mathematics that governs it. To understand how this works, imagine you are baking a large, delicious chocolate chip cake, but you do a terrible job of mixing the batter. All the chocolate chips end up clustered on the left side of the baking pan, while the right side is just plain vanilla batter. When the cake is baked and you cut it right down the middle, the slice on the left is packed with chocolate, while the slice on the right has absolutely none. Because of this unequal distribution, the two slices have completely different flavors and textures, even though they came from the exact same pan. In the fascinating world of developmental biology, a very similar process happens when a single egg cell divides. Now that we understand the basic idea through our cake analogy, let us look at the formal biological mechanism. Cytoplasmic determinants are specialized maternal molecules, typically messenger RNAs and proteins, that are localized to specific regions within the cytoplasm of an unfertilized egg. When the fertilized egg, or zygote, undergoes rapid cell divisions known as cleavage, the cytoplasm is not mixed. Instead, it is partitioned into new, smaller cells called blastomeres. Because these determinants were unevenly distributed in the original egg, the newly formed cells inherit different types and amounts of these molecules. These inherited molecules act as powerful signals that activate or repress specific genes within the nucleus, dictating whether the cell becomes a muscle, nerve, or skin cell. Understanding this mechanism is absolutely critical because it is the very first step in breaking the symmetry of a perfectly round, uniform egg. Without these localized molecules, every single cell produced during early division would be exactly identical, making it impossible to build a complex organism. In real-world medical and biological research, studying these determinants helps scientists understand how birth defects occur when early embryonic patterning goes wrong. Furthermore, this concept is foundational in stem cell research and cloning. Scientists must understand how a cell's internal cytoplasm programs its nucleus to adopt a specific identity, which could eventually allow us to reprogram cells to replace damaged tissues in human patients. While these determinants are biological molecules, their distribution across an embryo can be beautifully described using mathematics. Often, these determinants form a concentration gradient, meaning they are highly concentrated at one end of the embryo and gradually fade away as you move toward the other end. We model this exponential decay using the formula C of x equals C naught times e to the power of negative kx. Here, C of x represents the concentration at a specific distance x from the source. C naught is the initial, maximum concentration at the very origin. The constant e is the base of the natural logarithm, and k is the decay constant, which tells us how quickly the determinant degrades. If k increases, the concentration drops off much faster. Now that we have our formula, let us see this with a concrete example. Imagine a fruit fly embryo where a famous cytoplasmic determinant called bicoid is concentrated at the anterior, head end. We are given an initial maximum concentration of 100 units, a decay constant of 0.05, and we want to find the concentration at a distance of 20 micrometers. We start by substituting these values into our exponential decay model. The exponent becomes the product of negative 0.05 and 20, which simplifies to exactly -1. We then evaluate e raised to the power of -1, which is approximately 0.3679. Multiplying this by our initial concentration gives us roughly 36.79 units. This specific concentration will dictate the cell's unique developmental fate. This calculation we just performed leads us to an important biological concept: gene activation thresholds. In the same fruit fly embryo, the bicoid determinant acts as a switch to turn on a specific gene called hunchback. However, the hunchback gene will only turn on if the bicoid concentration is strictly greater than a specific threshold, which in this case is 15 units per cubic micrometer. This is a classic threshold problem in developmental biology. By finding the exact physical distance where the concentration drops to exactly 15, we can understand exactly where one body segment ends and another begins. Let us derive this boundary mathematically. To find this exact boundary, we are setting out to isolate the unknown distance variable, x. We start by setting our target threshold concentration of 15 on the left side of the equation. The right side remains our standard exponential decay formula, with the initial concentration of 100 and the decay constant of 0.05. To isolate the exponential term, we divide both sides of the equation by 100. This gives us 0.15 equals e to the power of negative 0.05x. This step tells us intuitively that the threshold is exactly 15% of the maximum concentration, making the subsequent mathematical steps much easier to handle. Now that we have simplified the equation, we need to bring the variable x down from the exponent. To do this, we take the natural logarithm of both sides. The natural logarithm is the perfect mathematical tool here because it perfectly cancels out the base e on the right side of the equation. This is a standard algebraic trick used whenever we need to solve for a variable trapped in an exponent. By applying the natural logarithm, we transform a complex exponential relationship into a simple linear one. The equation now reads: the natural logarithm of 0.15 equals negative 0.05 times x. We are now in the final stages of our derivation. Using a scientific calculator, we find that the natural logarithm of 0.15 is approximately negative 1.897. We substitute this back in, giving us a simple linear equation. The negative signs on both sides indicate we are on the right track, as physical distance must be positive. Dividing both sides by negative 0.05 gives us a final distance of 37.94 micrometers. This physically means that any cell located between 0 and 37.94 micrometers from the head will activate the hunchback gene. This sharp mathematical boundary creates complex biological patterns. So, we have proven how the mathematical boundary works. This brings us to a visual summary of the entire process. In this diagram, look at the top section first. You can see the original egg cell with red dots representing the cytoplasmic determinants clustered on the left. When the first cleavage occurs, the cell divides in half. Notice how the left cell inherits all the determinants, while the right cell is completely empty. Now, look at the graph below it. The vertical axis represents the concentration of these determinants, and the horizontal axis represents the distance across the embryo. The curve shows the exact exponential decay we just calculated, starting high at the source and dropping off rapidly. We have covered a lot of ground today, so let us review the most important ideas. First, Cytoplasmic determinants are the fundamental maternal instructions prepackaged into an egg before fertilization even occurs. Second, by distributing these molecules unevenly, the mother ensures that when the egg divides, the resulting cells inherit completely different chemical environments. Third, these different environments act as genetic switches, turning specific genes on or off and locking the cells into distinct developmental pathways. Finally, whether you think of them as flavor chips in a cake or model them mathematically as an exponential decay gradient, these determinants are the ultimate architects of early embryonic life. You now have a solid understanding of this foundational topic.